Hello everybody, welcome to your first MCDB video tutorial. Today we're going to do a brief Gen Chem review and an intro to macromolecules. You'll want to use the worksheet that I linked to this presentation to take notes, follow along. You'll get a lot more out of it if you do it that way. Okay, let's get started. So first up, a little bonding review. And first question we want to address is what are the main elements that organic and biosystems are composed of? That's going to be carbon, hydrogen, oxygen, and nitrogen. Now, why these four? Why are these the most prevalent? Well, these are the ones that form stable covalent bonds. They can form double, in other words, pi bonds, and they can form long chains, especially carbon in this case. So that makes them useful building materials. Now, the stability and ability to form pi bonds, these are stability and versatility reasonings. And carbon being able to form long chains, this allows us many building block possibilities, right? Infinite possibilities of putting things together. Okay, let's review the bonding schemes for these big four elements. So let's stick them here. So first off, hydrogen, as you know, will form one covalent bond. Carbon has the ability to form four, right? And these could be multiple bonds, double and triple. We're just showing single here in the structure for simplicity. Oxygen forms two bonds normally and has two lone pairs. So it does have a complete octet. So two bonds, two lone pairs. One thing you'll want to remember, though, is that oxygen can actually make a third bond. So it can use one of those lone pairs to, say, attach a hydrogen. Now, if it does that, it will take on a positive formal charge. And you've seen this before, like in acid-base chemistry, we had the hydronium ion, H3O+. Right, so the fact that oxygen forms two bonds, that's really in relation to it having a neutral formal charge. When we get to nitrogen, nitrogen normally makes three bonds and has one lone pair. But like oxygen, nitrogen can use its lone pair to make a fourth bond. But if it does that, it will take on a positive formal charge, as in ammonium ion, like we saw in Gen Chem acid-base chemistry. And we will see positively charged nitrogens quite a bit in our biochem journey this quarter. So something to look out for. Okay, we're going to finish up this section by talking about the two categories of bond types that we're going to deal with this quarter. And the first one is covalent bonds. And in biochem molecules, these bonds are about 50 to 110 kcals. This doesn't mean anything a priori, but this is an indicator that this is a strong type of bond, so strong that it's going to require help to break it. Right? So enzymes are going to have to be involved to break covalent bonds. The other bond type that we're going to deal with are weak bonds and forces. And they are much weaker than the covalent. They range from about 1 to 7 kcal, so you can see big difference there. And there's four types of weak bonds we're going to consider. hydrogen bonds, ionic bonds, hydrophobic forces, and van der Waals forces. And to make biochemistry work, we need both the weak bonds and forces and the covalent bonds. All right, more on the weak bonds and forces in just a minute, but before we get there, we want to review functional groups. So these are the little working groups that are reactive on the particular molecules. So here we have a biomolecule, threonine, and we're going to use it to illustrate some of the functional groups. So here we see a hydroxyl group, an OH group, which is a polar group. Here we have illustrated an amino group. Now, amino groups are basic groups. You can see the lone pair wasn't drawn in on the initial structure, but it has the capability of picking up a proton, acting as a base, and therefore taking on a positive formal charge. And this is quite common. The other functional group of note in threonine is the carboxyl group. And the carboxyl group is an acidic group. You can see that we've lost the proton here and taken on a negative charge. So this oxygen actually has 3 lone pairs, which gives it a negative formal charge. We'll see that quite a bit as well. This is actually an example of an amino acid named after its 2 main functional groups. Here's another example for us to look at. This is coenzyme A. It's involved in metabolism processes. And you can see as we inspect this, you can see there's a couple of hydroxyl groups hanging off there, as well as another amino. But also illustrated here is a phosphate group and a sulfhydryl group, sometimes called a thiol. Right? So these are all important reactive centers on this molecule. All right, let's go back to our weak bonds and forces and overview them briefly. So first up, we're going to talk about van der Waals forces. And you studied these in Gen Chem under the name of London dispersion forces, or LDFs. So remember what that was about. These were attraction between induced and temporary dipoles. So any 2 molecules can be attracted to each other through temporary dipoles. Right? So anything has the possibility of LDFs. However, they're very weak. They're on towards the 1 kcal end of the spectrum in weak forces. It is worth noting that the strength of the LDFs actually increases with the number of electrons on the molecule. And so if you have a pretty large molecule with a lot of electrons associated with it, it's actually easier to induce the dipoles and thus stick to something else. Right? So these can be significant and add up. Next one we want to touch on is a hydrogen bond. And a hydrogen bond is really a specialized dipole interaction that involves an N and an O and an H. It can also involve a fluorine as the electronegative atom, but we really just don't see those in the biochemical systems. But technically, fluorine can be a player as well. So you can see here we have a dipole set up between this nitrogen and hydrogen in the covalent bond. Nitrogen is electronegative, so this sets up a delta negative here and a delta positive here. And that is then attracted by this electronegative oxygen, right? So here's the actual hydrogen bond. One thing that's interesting is that they're the strongest when the 3 atoms are in a straight line, like they're shown here. And here's a couple of more examples. So here's some more illustrations of hydrogen bonds. This is in a polypeptide chain or a protein. We have protein backbones here, and you can see an interaction between the O on one chain and the nitrogen-hydrogen group on the other chain, and another one here. Another place where hydrogen bonds come up are in nucleic acids, in DNA or RNA. This is showing DNA, where we have hydrogen bonding here. Okay, now these were all showing the hydrogen between an O and an N. It can be any combination of electronegative atoms. So you could have 2 O's with a hydrogen in the middle, 2 N's with a hydrogen in the middle, or like the combinations you see here with an O and an N and an H. Next one is hydrophobic forces. Hydrophobic is another word for nonpolar. So this is when nonpolar groups aggregate to avoid water. Remember that old saying, like dissolves like? All right, well, that's what's going on here. Things that are similar want to be together because they have similar interactions. The way to identify a nonpolar group is to look for hydrocarbon groups. So hydrocarbons are considered nonpolar. So you can see here, these are little water molecules all. We have a couple of methyl groups stuck in here. So 2 nonpolar groups, that's pretty uncomfortable. So to avoid the water, they're clustering together, right? So that's a hydrophobic interaction. Here's another example. Here's 2 hydrocarbon groups clustering together to avoid water. Next up is ionic bonds. And of course, an ionic bond is when you have opposite charges attracting. That's pretty straightforward. What might be bothering you is, why are we putting ionic bonds in this category of weak bonds? When we study this in Gen Chem, we were studying it as ionic bonds being really strong, and compounds that had ionic bonds were in fact solids at room temperature. And that's not incorrect, but in biochemistry, it's all about the context. In biochemistry, everything is in aqueous solution. So we have to look here at what's going on in aqueous solution. So here we have our charges that are attracted, but that attraction is dissipated by the waters getting in between. So that makes it now a weak force, a weak bond. All right, so all about the context there. Ionic bonds are actually weak in aqueous solution. Okay, let's take some time now and do some application and see what we've learned. So pause your video and try out these questions. All right, let's see how you did. So how many hydrogens did we need to complete that structure? Well, this is based on the fact, what? That carbon makes 4 bonds, oxygen makes 2 bonds normally, right? So you should have come up with 10. And if I put the hydrogens on, this is where they go. You'll see there's 10 of them. In case you're not familiar with this kind of drawing, it's understood that a carbon is here at each of these points. All right, so here we have a carbon. It initially just had 3 bonds, so we needed to have a hydrogen to bring it up to 4. This oxygen just had 1 bond, so we needed a hydrogen to bring it up to 2, etc. Next one, which of the following is not a correct example of hydrogen bonding? You should have picked C there. So if we check this out, here's the hydrogen, and you can see it's between an oxygen and a carbon. A carbon is not electronegative enough to set up or receive a hydrogen bond. You need an O or an N here in this position. So you can see the other ones are good. We have the hydrogen between an O and an N here, a hydrogen between 2 N's, so that's okay, and a hydrogen between 2 O's here. Here the carbon is farther away, so it's not involved. Next question, which of the following will be least soluble in water? So water's polar, so the least soluble one will be the most nonpolar. And remember how we find nonpolar groups? Nonpolar groups are hydrocarbon groups. So these are actually 3 amino acids. So you can see they have the same structure going across here. And what's different about them is what's hanging down. All right, so if we compare that, you can see what? This one here has the biggest hydrocarbon group, and therefore that is the most nonpolar and therefore the least soluble in water. All right, let's finish up today with a brief macromolecule overview. So macromolecules are composed of subunits linked together via what kind of reaction? That's right, a condensation reaction. This is also known as a dehydration reaction because water, in most cases, is one of the products. So if we take a couple monomers here, we can split out a water and link the 2 dangling pieces together to create this dimer. All right, so we've new covalent bond, we've created water. And to make the macromolecule, we just repeat this. So we bring in a 3rd monomer, split out a water, link it in the chain, etc., as many times as you want for as big as you need. All right, what are the classes of macromolecules and what are the subunits that they're formed from? So if we take glycerol and fatty acids, that's how we make lipids. If we link together amino acids, we make proteins. If we take simple sugars like glucose and link those together, we get polysaccharides. And finally, if we take nucleotides and assemble them in a chain, we get nucleic acids, RNA and DNA. Okay, so that's it for this session. We'll talk more about macromolecules next time, more specifics. Until then, happy studying.